Biology 1107, presentation number 12, Genetics. Genetics is the study of the inheritance and the expression of traits. There are two forms of genetics, or two subsets of this discipline. One's called Mendelian genetics, and the second is called molecular genetics. They differ in age by about 100 years. Give or take. Mendelian genetics dates back to the work of a fellow by the name of Gregor Mendel, a monk in Austria. And we'll talk a little bit more about him, but he published a paper in 1865 that was largely ignored. But in this paper, he set out a set of laws that governed the inheritance and expression of traits at the level of the individual. And is extremely watershed publication. It was discovered after his death and republished in the early 1900s and really established the tone for genetics of today. As a consequence, Mendel is known as the father of genetics. Now, molecular genetics hails its origin to the publication of the structure of DNA by Jim Watson and Francis Crick in 1953. And in that particular paper, they not just described the structure of DNA, but proposed that DNA molecule as a mechanism of encoding information, a mechanism of inheritance, and ultimately a mechanism of expression traits. And as time has gone by, researchers have determined the details of how that information is encoded, and we now understand that that encoded information is used for the purposes of producing proteins, and we understand that those proteins are ultimately responsible for all of the characteristics of life, what we would call traits. And so therefore we see a connection between what is molecular genetics and Mendelian genetics. Now, in our presentation today, we're going to be covering Mendelian genetics, the inheritance and expression of traits at the level of the individual. We will go into greater detail about molecular genetics on a couple of presentations not too long from now. But right now, we will be focusing on Gregor Mendel and his laws that govern the inheritance and expression of traits at the level of the individual. And then what we'll do is we'll take a look at our understanding of those laws and how they apply to our understanding of genetics today. Gregor Mendel was a monk living in a monastery in Austria in the first half of the 1800s. And his job at the monastery was to tend the monastery garden. He was responsible for growing the vegetables and actually providing a substantial amount of food for the monks at that monastery. But in spare time, what he did was he bred garden peas. He recognized that garden peas had pretty flowers and they came in different colors, and he recognized that garden peas had traits of seed color and seed shape, and as a matter of fact, how tall the plants grew. So in his time spent at the monastery, he bred garden peas and took notes. And in doing so, he came up with some realizations about the inheritance of traits. In his garden peas, he recognized certain patterns, and he summarized these patterns in what we now know as Mendel's laws. There are 4 different laws. Now, the story of Mendel basically culminates in his publication of a paper in 1865, and I mentioned it not too long ago, and I said it was largely ignored. On the recommendation of his colleagues in the monastery, they said, look, you're spending a lot of time doing this, and you're coming up with a tremendous amount of results, and from what we can see as valuable results, why don't you publish this information in a paper? Submit that paper to the local scientific society, and they'll publish it, which he did. And it was published. But like I said, it was largely ignored. And that didn't bother him at all. He just went back to doing his garden pea thing and tending the garden, and it wasn't too much longer after that that Mendel died. Now, about the same time, this fellow by the name of Charles Darwin comes up with this theory of evolution by means of natural selection. The idea is that there's conditions in nature that select for certain traits, that select for certain adaptations in organisms to improve their ability to survive and reproduce in the environment in which they live. So, in other words, Darwin establishes his theory that explains why it is that organisms seem to be so well matched or adapted to their individual environments. But the problem with this theory of evolution is it did not have a mechanism. At least at that point, folks had a rudimentary idea that traits were passed on from generation to generation to generation to generation, and traits seemed to characterize certain species, but there was no understanding of genetics, what we kind of take for granted today. We know that traits are encoded in our genes, and we know if those genes are selected for, they will be passed on to future generations, and that will guide what future generations of a species might actually look like. But the problem in the 1850s is there was no mechanism to support this. Now, admittedly, Mendel's work provided that mechanism, but his work, published in a rather obscure scientific journal, was not read nor appreciated by a lot of folks, primarily because at that point, Darwin's theory, Darwin's theory was published in 1859, it'd only been out there for about 6 years. So really not a tremendous amount of time for people to kind of mull on it and realize that there has to be some kind of mechanism to support Darwin's notion about evolution by means of natural selection. To make a long story short, Mendel dies. The paper kind of lapses into obscurity until some scientist in the early 1900s finds it. He finds it. I don't know if he finds it in a box at a yard sale or what, but he finds the paper. He reads the paper. He shares it with his friends. And together, they recognize the fact that this is, and I use the term watershed, this is a watershed paper, because they realized that this really presented the mechanism, the basis for how Darwin's evolution theory could actually operate. And so they realized it was worth folks to read, and so they republished it. They republished the paper in Mendel's name. It was published, I guess, probably about 50 years after his death, at which point everyone read this paper, and all of a sudden it propels a bunch of folks to study what we would now call genetics, the understanding of how traits are inherited and expressed, because our understanding of how traits are inherited and expressed lends all kinds of meaning to Darwin's theory of evolution by natural selection, which at this point in the early 1900s was going gangbusters. So Mendel at that point becomes known as the father of genetics and largely appreciated for what he did. And we're going to document just how successful the man was. We're just going to remember that Mendel had no formal scientific training. He had no laboratory. He had no scientific equipment. And yet what he did was of just tremendous import, and really everything we know today, not just about genetics, but a large part of what we understand about molecular genetics owes its existence to him. Here is our bird's eye view of Mendel's laws. These are his recognition of patterns demonstrated in his garden piece. Now, admittedly, the garden piece is just one small organism out of a world of thousands and thousands and thousands of organisms. To be able to generate some laws that have some meaning on the basis of just one sample organism is pretty amazing. And what's also amazing is, like I said, he had no formal scientific training. He had no laboratory. And this was just kind of like an offshoot of his own personal hobby. So what we're going to do is we're going to document the 4 laws here. The law of unit factors, the law of segregation, the law of dominance, and the law of independent assortment. I'm going to discuss each one, and as I do, I'm also going to mention essentially how Mendel did in terms of these laws, because obviously he had no clear idea of how appropriate they were to biology in general. Matter of fact, I'll just go ahead and do that now. Law 1, law of unit factors, he was 100% correct. What he found in his garden peas has application to every animal, every plant on the face of the planet. Item number 2, law of segregation. He was 100% correct on that. Has application to every single animal, every single plant on the face of the planet. Law number 3, law of dominance. Mendel didn't quite get it all right here. So we can say that basically he was maybe 1/3 of the way there. If I said he was a baseball player, he was batting .333 on the law of dominance. Now, as baseball players go, that's a pretty darn good batting average. So even though he was 1/3 correct, Mendel really was doing rather well at this point. Now, the only law he really messed up on was number 4, the law of independent assortment, and he completely struck out on that one. So now that we know how well he did, let's discuss what these laws actually mean. The first law of unit factors is a law that essentially examines what we now call genes. Now, please understand that back in the 1850s, there was no term genes. So in the absence of having a term, Mendel called these things unit factors. So unit factors are what we would call genes today. And his understanding was parents, that is a pea plant that was going to be bred with another pea plant, would have 2 unit factors each. So, for example, if you've got a guy named Joe and he marries a girl named Sue, he's got 2 genes for eye color, and so does Sue. Because the fact of the matter is he has inherited a gene from eye color from his mom and a gene for eye color from his dad, and same thing with Sue. So parents have 2 each, and one of those unit factors, or what we call them genes today, actually code for one trait. So in his pea plants, he might breed a red pea plant with a white flowered pea plant, and the red flowered pea plant has 2 unit factors for flower color, and the white flowered pea plant has 2 unit factors for flower color as well. Now, item number 2, law of segregation. There was no understanding of meiosis in the 1850s, at least from Mendel's standpoint. There wasn't. I mean, folks had recognized the existence of meiosis, but it was fairly rudimentary. But he comes up with this law of segregation. Now, the law of segregation means meiosis. The idea is this. If you've got a parental type, a red flowered pea plant, or a white flowered pea plant, he knew that each of those pea plants had 2 copies of their unit factors, right? What we call 2 copies of genes. Each of those parents is going to pass only one of those 2 on to the offspring. Now, you and I both know at this point that meiosis is a specialized form of cell division that takes a diploid cell, that's a cell with 2 copies of each gene, and creates a haploid cell, a cell with only 1 copy of each gene. So he has no formal idea of meiosis, but in his peas, he recognizes that parents having 2 unit factors for any given trait are going to pass one of those on to the offspring. So 2 reduced to 1 is essentially what he calls segregation, is what we would call meiosis today. Okay, so we've been through Law 1 and Law 2. He's 100% correct on law of unit factors. He's 100% correct on law of segregation. What about the law of dominance? To understand the law of dominance, what we have to do is understand what Mendel's concept of dominance was. For many of us, we think of dominance as something that we can establish a value to. In other words, dominance is good, as opposed to being recessive, which is bad. No, that's not the way it is. Dominance for Mendel was the tendency of a trait to be expressed. A dominant trait tends to be expressed, whereas a recessive trait tends not. It has nothing to do with the actual benefit or the value of the trait. It has everything to do with whether it tends to be expressed or not. So when he took a look at his garden peas, he recognized that if an individual inherited two unit factors, one from one parent and one from the other, of those two inherited unit factors, only one of them is going to be expressed. So for example, if he had two parental pea plants, one red flowered and one white flowered, and if the red flowered pea plant passed on a red flowered unit factor to its offspring, and the white flowered pea plant passed on a white flowered unit factor to the offspring would be red, meaning that the red offspring was actually expressing the unit factor inherited from the one parent. So only one inherited unit factor will be expressed. That's the law of dominance. Now, a little bit later, we will find there are actually a couple of exceptions to that. Mendel's original concept of dominance is sometimes also called simple dominance. So you might want to scribble that next to this particular law here, simple dominance. On a future slide, we will document the other two variations on this theme. About the only thing I can say is, yes, this is one of the limitations of basing your results on a single sample organism. Garden peas demonstrate simple dominance. They do not demonstrate any other variations. However, there are plenty of animal species and plenty of plant species that demonstrate other patterns besides this. And that's why Mendel gets the 1/3 correct on law number 3. Now, the last one is the law of independent assortment. And this law states that any one trait is inherited independently of any other trait. In his garden peas, flower color was inherited independently of seed color or seed shape. He could not connect a relationship between those because apparently those traits were inherited independently. But nowadays, we do know that these traits are found on chromosomes. And if two traits are found on the same chromosome, those two traits move together. Wherever that chromosome goes, those two traits go together. Which means if those two traits wind up moving with that chromosome into a brand spanking new individual, those traits will be inherited together. Those two traits are said to be linked. So the law of independent assortment, at least as far as Mendel was concerned, is the idea that traits are not linked. But in fact, today we do know they are linked because if traits occur on the same chromosome, wherever that chromosome moves, those linked genes will move together. So quick question, Knaus. Thing is, if all organisms have their genes found on chromosomes, then why is it that Mendel didn't see linkage in his garden peas? Because if those traits are found on chromosomes, I would have thought that he would have actually observed linkage in those traits. Well, the only explanation I have is perhaps in his garden peas, those traits he was looking at, flower color, seed shape, seed color, plant height, those different traits may have actually been located on different chromosomes. If they were located on different chromosomes, they wouldn't necessarily demonstrate linkage. But by and large, most traits do demonstrate some degree of linkage with other traits because they are found on common chromosomes. So in this case, yes, Mendel strikes out, but he strikes out because primarily, once again, he only used the garden pea as his representative study organism. Tell you what, not bad for a guy with no formal scientific training, working out of a monastery in the middle of Austria in the middle of the 1800s. The man did a tremendous amount and has earned every bit of fame he's got. So now that we've taken a look at these laws in context of Mendel's work at his time, let's see what we understand today. Now, today we see that unit factors are what we call genes. A gene, here's a definition which you've heard before, and it won't be for the last time. A gene is a sequence of DNA that codes for a protein. A protein is responsible for a trait. So we've got our genes arranged in pairs, and all sexually reproducing organisms have their genes arranged in pairs. Animals have their genes arranged in pairs. Plants have their genes arranged in pairs. Now, an individual copy of a gene is called an allele. So we have, each of us, 2 alleles for eye color. We have 2 alleles for every other gene found in our collection of chromosomes. And so alleles can, as a rule, come in 2 different flavors. Alleles can be dominant or alleles can be recessive. Now, I will remind you the word dominant implies this is an allele that tends to be expressed. A recessive allele tends not to be expressed. Has nothing to do with any valuation that you can place on the allele. Has nothing to do with anything but whether it tends to be expressed or not. Give you a basic idea. Eye color, I'm going to be very simplistic here. If you got brown eyes, an allele for brown eye color is a dominant allele. The allele for blue eye color is a recessive allele. Now, if you inherit 2 brown alleles, you will have brown eyes. If you inherit 2 blue alleles, you'll have blue eyes. But if you inherit an allele from brown eye color from one parent and blue eye color from the other parent, you will still have brown eyes. In that case, the brown eye allele, the dominant allele, tends to be expressed, whereas recessive tends not to be expressed. Now, we also know that these alleles that we're talking about are actually found on chromosomes. Mendel was not aware of chromosomes, but we do know that we have these things called chromosomes. Chromosomes are our little genetic suitcases. It's how we move traits from one cell to another cell or from one generation to another generation. We can actually map where alleles are located on chromosomes. The specific location of an allele is called a locus, or loci, plural. A locus is the actual physical location where an allele will occur on a chromosome. Then finally, 2 terms that you will need to know. I guarantee you they will show up on questions. And if you don't know the meaning of these terms, you're going to be hopelessly lost. So here's what I need you to know. Genotype is a word that means the allele combinations that an organism has. The allele combinations that an organism has. For example, talking about eye color, suppose you've inherited a dominant allele from your dad and you have inherited a recessive allele from your mom. You've got one dominant and one recessive. That is your genotype. It's the allele combination you have. Another way of thinking about it is I usually think about it this way. I say genotype is what my genes say. I've got one allele for brown eyes. I've got one allele for blue eyes. That's what my genes say. That's my genotype. It's my allele combination. But in contrast, we got another word here called phenotype. And phenotype is the expression of genotype. So if you've got an allele for brown eyes from one parent and an allele for blue eyes from the other parent, but if you look in the mirror, you've got brown eyes. Brown eyes is the expression of your genotype. Brown eyes is your phenotype. So phenotype is the expression of genotype. And If I said that genotype is what your genes say, well, then phenotype is what your genes show. Well, let's take a look at the relationship between genotype and phenotype. We'll take a look at genotype first. Remember, we said that there were two possible gene states for any one gene. Said genes come in two different flavors. I said some are dominant and some are recessive. So here I see there are three basic possible combinations. If your genes come in either dominant alleles or recessive alleles, then there are basically three possible combinations. If you only have space for two, either you'll get two dominants, two recessives, or a dominant and recessive. Now, there are going to be some terminology that goes along with this. So you do need to know the terms genotype and phenotype. Well, there's some other terms here you will need to know, and they're going to be critical to your understanding of any question that might be posed to you on an exam. Take a look under genotype. You could conceivably have inherited two dominant alleles. We're going to signify a dominant allele with a capital letter. In this case, A symbolizes whatever gene we want it to be. It could be eye color. It could be flower color. But because we use the uppercase A, it's symbolic to us to be a dominant allele. And if you inherit two dominant alleles, one from each parent, you are referred to as being homozygous dominant. That word homozygous comes from the fact that when you are created from a zygote, remember a zygote is a fertilized egg, that zygote contains the very first two alleles. And if you got two dominant ones, you have two of the same in the zygote. So big A, homozygous means two of the same in the zygote and two of the same dominant alleles in the zygote. If you have two recessives, little a, then you are said to be homozygous recessive. If you have one of each, they're different, so we're going to say that's the heterozygous genotype. So genotype is essentially described by three possible terms here. Homozygous dominant for big A, homozygous recessive for little a, and heterozygous for big A, little a. Now, those genotypes are going to result in an expression which we call phenotype. Now, following Mendel's original idea, remember I said that was called simple dominance? And he said of the two unit factors present, only one will be expressed. Well, now today we would say of the two alleles present, only one will be expressed. And in our heart of hearts, that's going to be the dominant allele. So in a big A, or homozygous dominant genotype, there's two big A's, so I have no choice. You will be a dominant phenotype. If you're a homozygous recessive, little a, you are obviously going to express the recessive phenotype. But if you're big A, little a, as long as you got that dominant allele there, you will express the dominant phenotype. So the little disclaimer at the bottom of the slide is that this is typical of what we call simple or sometimes called complete dominance. This is Mendel's original concept. It says it is not typical of codominance, which we'll see in a moment, nor is it typical of incomplete dominance, which we will also see in a moment. So there are three versions of dominance here. Simple dominance, which we're looking at right now, codominance, and incomplete dominance, which we'll see in a little bit. Well, how about that, Dr. Knauss? I see on this particular slide you have simple dominance listed, codominance listed, and incomplete dominance listed. And I suppose what you're going to do is you're going to show me how those dominance patterns vary. Okay, so we're going to assume flower color in this case, and we're going to say the dominant allele, uppercase A, codes for red flower color, and the recessive allele, lowercase a, codes for white flower color. Now, in an example of simple dominance, we would have three possible genotypes. Big A, homozygous dominant, little a, homozygous recessive, or big A, heterozygous. Now, as long as a dominant allele is present, the plant will have red flowers. Hence, big A has red flowers, big A has red flowers, and the only case we'd have white flowers is little a, because there is no dominant allele to outshine the recessive allele, so to speak. In codominance, what we're going to do is we're going to focus on the heterozygous situation. That's the big A, smack dab in the middle. So here's the deal. In codominance, it states that if both alleles are present, meaning if both a dominant allele and recessive allele are present, both will be expressed. If both alleles are present, then both will be expressed. So let's suppose I'm breeding petunias. Petunias are flowers, and if I breed a petunia and it's big A, that petunia is going to have red flowers. And if I have a petunia that's little a, that petunia is going to have white flowers. But now, if I have a big A, little a petunia, that petunia is going to have red and white flowers. In other words, the alleles are expressed together. They're completely expressed. So as long as I've got a red allele and a white allele, my flowers are going to be red and white. That's codominance. Now, the last example here is incomplete dominance, and we're just going to phrase this just a little bit differently. Once again, I'm going to be focusing on the heterozygous genotype, big A, little a. In this case, we'll say that if both alleles are present, then they will be incompletely expressed. If both alleles are present, meaning the dominant allele and the recessive allele, if both alleles are present, then they will be incompletely expressed. So let's see, incomplete, that's kind of like halfway between white and red, Dr. Canal. So what's halfway between white and red is pink. So if we are a big A petunia, I'm going to have red flowers. But in this case, if I'm big A, my flowers are going to be pink. And then if I'm little a, then my flowers are going to be white, as would be typical for a homozygous recessive. So be aware of the fact that there are these three different dominance patterns: simple dominance, which Mendel was correct on; codominance, which Mendel didn't even know existed; and incomplete dominance, once again, something Mendel had no idea of. But nowadays, we do know that the difference between simple dominance, codominance, and incomplete dominance boils down to that heterozygous genotype. And what is the phenotype that results? Is it the dominant phenotype? Is it part dominant, part recessive? Or is it kind of an in-between? So we've revisited unit factors. We understand those are genes, also known as alleles. We revisited segregation, and we know that is meiosis. We revisited dominance. We understand there's actually three patterns of dominance. And let's revisit independent assortment, the idea that any one trait is inherited independently on any other. However, we understand there are these linkage groups where these traits might be located on a common chromosome. So if several traits are all found on chromosome number 1, wherever chromosome number 1 goes, so will all those traits move together. Now, you'll notice on this slide, I've got a bullet point listed X-linked traits. There are some traits that are all found on the X chromosome. Now, the interesting thing about the X chromosome is it's critical in gender. If you're a female, you're XX. If you're a male, you're XY. Now, it turns out if you're a male and you inherit an X chromosome that has an X-linked trait on it, you will express that trait. So, for example, color blindness is called an X-linked trait because it's found on the X chromosome. Wherever the X chromosome goes, so will that color blindness allele. Now, if you're a guy, you inherit your X chromosome from your mom. So you inherit potentially a color blindness allele from your mom. You inherit your Y chromosome from your dad. But the combination of the X and Y means there is nothing on the Y that's going to keep the X from being expressed, which means if you inherit that color blindness allele on the X chromosome, you will express color blindness. Color blindness is an example of an X-linked trait, a trait that is connected to the X chromosome. But there are plenty of others, but those are found on chromosomes number 1 through 22, and those, as a rule, are not designated as anything but a form of linkage. But we do know that in all organisms, linkage is a thing because all of our genes are located on chromosomes, and all genes on a common chromosome will all move together as a linkage group. On this slide, we're going to delve a little bit into some additional information we know today, something that goes above and beyond what Mendel knew and above and beyond what some of the early genetics researchers knew as well. Let's start off with our first two bullets, pleiotropy and polygenic inheritance. These two bullet points indicate something we know today that scientists did not know 100 years ago. So let's start off with what scientists knew 100 years ago. What they knew was largely based on what Mendel knew. And Mendel knew that a unit factor, what we now know is a gene, governed one trait. This is commonly known as the one gene, one trait concept. And as a matter of fact, the far majority of genes code for one trait. Now think about it. What this means is one gene codes for one protein, which codes for one trait. So what could be in defiance of that? Well, pleiotropy for one, and then polygenic inheritance. Let's talk about pleiotropy. I'll define it first. I need you to make sure you write down that pleiotropy is where one gene influences several traits. One gene influences several traits. What this means is one protein could conceivably have an effect on several different traits. So how about an example? I will give you an example, and as an example from American history, and it involves Abraham Lincoln, and it involves a genetic disorder that is a mutation that results in something called Marfan syndrome. M-A-R-F-A-N. Now, Marfan syndrome is a consequence of a mutation of a single allele on a single chromosome. That means is you are producing a protein that is going to have an effect on several different traits that will result in this thing called Marfan syndrome. Let me explain. Abraham Lincoln. Abraham Lincoln is our first known person to have Marfan syndrome, even though Marfan syndrome was not described when he was alive. So here's the basic idea. Abraham Lincoln was this tall, gangly guy. That particular form of body is commonly referred to as body habit. He had a long, tall, lanky body habit. And not everyone who has a long, tall, gangly body habit has Marfan syndrome, but it is a manifestation of that syndrome. So one allele resulting in one protein, results in this tall, gangly body habit. So what else, Dr. Knaus? Okay, well, Abraham Lincoln. You take a look at pictures of Abraham Lincoln, or you even read the memoirs of his secretary, a fellow by the name of Nicholas Hay. He documents the fact that during his second term, Lincoln was losing his eyesight. Photographs show you that Lincoln starts wearing glasses. And according to his secretary, it wasn't long before Lincoln was about as blind as you could possibly be. And that was all happening during his second term. And it's a documented effect of this particular syndrome, Marfan syndrome. So not only does this protein cause an influence on a person's body habit, but it also has an influence on a person's eye health. And then thirdly, when Abraham Lincoln was autopsied after his assassination, medical doctors determined that his heart was in very, very bad shape. So heart deterioration technically is a manifestation of Marfan syndrome. So here we got one gene, one protein, multiple traits. Now the flip side of that is, could it be that multiple genes might have an influence on one trait? And that's what we're going to call polygenic inheritance. Sometimes it's also known by the term polygeny, Polygyeny. It is the idea that multiple genes have an influence on a single trait. Let me give you an example. The example is skin pigmentation in humans. If you take a look at skin pigmentation in human beings, it's all over the map. We got folks who are pale. We have folks who have a yellowish skin. We have folks who have an olive skin color. We got folks who have brown skin color. We got folks who have a reddish skin color. We got folks who have a black skin color. It's all over the map, like I said. And obviously it does sort out with race. But the thing is, there is not one gene that influences skin pigmentation. It's more like a constellation of 5 to 7 different genes has an influence on skin pigmentation. It's one of the reasons why you have such a tremendous amount of variation. So pleiotropy, one gene has an influence on multiple traits. Polygeny, where several genes have an influence on a single trait, operate in defiance of the one gene, one trait idea. Now, the next two bullet points on our slide here, phenotypic plasticity and effects of the environment, kind of go together. I would have originally had put these as essentially subsets of the same bullet point, but Microsoft decided to arrange them as two distinct bullet points. So what you might want to do is link them with a double-headed arrow. Because the idea that essentially a gene and its result does not exist in a vacuum. So any gene and its result is going to exist in the context of an environment. Think about a tree. See, if I go out in the woods and I find myself a hickory tree, hickory trees are really big. And under that hickory tree, around on the bottom of the forest floor, I might find some baby hickory trees. Now, the baby hickory trees are just really tiny versions of the big, tall, giant hickory trees. But I say this, a baby hickory tree is growing in almost complete shade. It's not tall enough to be able to take advantage of the sunshine. So if I take a look at a baby hickory tree, one thing I note is the baby hickory tree leaves are like 3 times as big as the leaves found on a mature hickory tree. Now, the only explanation I can have for that is this is an influence of the environment on the leaves of that baby hickory tree. So obviously, the leaves of a baby hickory tree are programmed into the genes of the hickory tree. But lack of sunlight results in that basic plan, that basic leaf being 3 times as big as it ought to be. primarily for the point of absorbing more sunlight. So leaf size in hickories, as a consequence of light intensity, is a form of phenotypic plasticity. Now, the word plasticity means variation. Okay, it doesn't mean plastic in the sense of an organic polymer that's used for making things. Plasticity is variability, and phenotypic plasticity is the variability in phenotype which exists as a consequence of the environment in this example. Now, our next bullet point is the term epistasis. And epistasis is the idea that you could have a gene somewhere in your set of chromosomes. Maybe it's on chromosome number 15. And that gene on chromosome number 15 might have an influence on the expression of a gene on chromosome number 4. So it's the idea you've got a gene on a completely different chromosome having an influence on another gene on another completely different chromosome. That's epistasis. Then finally, we're going to talk about genes with multiple alleles, and we're going to use blood typing as our example. And we'll get into that in a minute, but this is the basic idea. For most genes, we already mentioned there are two possible forms, a dominant form and a recessive form. So we're talking about gene R, big R, little r. We're talking about gene A, big A, little a. However, in some cases, you might have a gene that doesn't just have a dominant and a recessive. It might have 3 different alleles. So, for example, in blood type, you and I, or any other American or any other person on the face of this planet running around is going to have 2 alleles for blood type. One that you inherited from your mom and one that inherited from your dad. But the thing is you might have type O blood, you might have type A blood, type B blood, or type AB blood. Now, the only way you could possibly get multiple blood types is if you have multiple alleles. Turns out, in the human population, there are type A alleles, there are type B alleles, and there are type O alleles. So instead of just being a dominant and recessive thing, I've got 3 different alleles governing blood type. And depending on which 2 alleles that you inherit from your parents, that determines whether you're A or B or AB or O. So it's not a simple case of dominant or recessive. Here's our detail slide on blood type. We understand that blood type is governed by 3 alleles in the human population, not just 2. The alleles are I superscript A, I superscript B, and lowercase I. Now let me explain the symbolism here instead of using just A, B, and O. The capital letter I indicates a dominant allele. In the case of blood type, I superscript A and I superscript B are codominant, as indicated by the genotype I superscript A, I superscript B below. Follow the arrow and you see that genotype codes for type AB blood. In other words, both A and B are fully expressed. Now, lowercase I, what we would consider to be the O allele, is recessive. And the only time you have type O blood is if you have 2 lowercase i's. Now, remember, when you get blood type in an individual, it is a combination of whatever 2 of those 3 alleles are inherited. So, fortunately or unfortunately, you need to know everything on this slide. You need to know the genotypes and you need to know the representative phenotypes. Now that we have examined blood type as an example of multiple alleles, we're going to set that aside just for a moment. We will return to it when we start talking about crosses. Crosses are matings. If you have 2 individuals with their unique phenotypes and their even more unique genotypes, perhaps we'd like to know what happens if those two individuals mate. What's our probability that we're going to have an offspring of a particular phenotype or maybe a particular genotype? So crosses are matings, and their purposes is to predict results or understand the details of the inheritance of specific traits. See, when Mendel was working with garden peas, he was doing crosses. He was doing crosses in real time. He was mating his peas and recording the results. Now, gotta let you know that it's possible to perform a cross on paper. You don't have to perform crosses in real time, particularly if you're talking about an organism that has a long reproductive life, and it might be a little bit difficult to get in a whole lot of study over one human's lifetime. So crosses can either be matings done in real time or they can be done on paper. They come in two types. There are monohybrid crosses and there are dihybrid crosses. A monohybrid cross examines the inheritance of one trait. Now, in a monohybrid cross, if we're following the inheritance of one trait, what we will do is we'll come up with 4 possible outcomes. In other words, of all the possible outcomes can arise from a single mating between a male and a female, there are 4 outcomes at 25% probability each. Hold that thought. We'll see it in a moment. In a dihybrid cross, what we're doing is we're looking at the inheritance of 2 traits. And those 2 traits give us 16 possible outcomes. So what you can see is that what's happening is every time we add another trait, the cross becomes a little bit more complicated. Now, I got to admit, there's trihybrid crosses out there, but they're pretty darn complicated. As a matter of fact, if I was to do a cross for a set of 3 different traits, I would have 64 possible outcomes. So most practitioners of genetics are going to want to keep things simple as possible. Now, I want to explain something about these crosses. Now, if we're going to examine the inheritance of one trait, it doesn't mean that our organism has only one trait. We just choose to focus on that. So, for example, when Gregor Mendel was breeding his garden peas, he might have been focusing on flower color. It doesn't mean that his plants don't have seed color or seed shape or plant height or any other characteristics. It's just that he chose to focus on the inheritance of flower color for those plants. That's a monohybrid cross. On the other hand, Mendel could have been focusing on flower color and seed shape. Those are 2 traits. And he would have come up with 16 possible outcomes. But it's because he decided to focus on just 2 traits, flower color and seed shape, and just chose to kind of ignore the rest of them. It just kind of makes life easy. So if I'm raising guinea pigs and I want that perfect guinea pig to get me that blue ribbon at the state fair, and I might be interested in coat color, and I might be interested in coat length, and I want to breed myself a platinum long-haired guinea pig because I know that is not only going to give me a blue ribbon at the state fair, but it's probably going to make me some money in breeding rights as life goes on. So how do I do a monohybrid cross? How do I do a dihybrid cross? Well, the steps are as follows. We need to determine the gametes or the sex cells to be formed. I got 2 individuals, a male and a female. And I know the phenotype of my male, and I know the phenotype of my female, and I might even know a bit about the genotype of my male and the genotype of my female, to the extent I might be able to predict exactly what type of sperm he's going to make or what type of eggs she's going to make. In other words, which alleles go into that sperm, which alleles go into that egg. Then what I'm going to do is construct and complete what's called a Punnett square, which is a tool for matching gametes and coming up with new combinations. If I have one sperm fertilizing one egg, Punnett square allows me to predict what the outcome's going to be. And then I can evaluate my results. Am I going to get that platinum long-haired guinea pig? I don't know. What's the probability I might get that platinum long-haired guinea pig? If I do this one mating, is it 75% chance? It's probably worth my while. If it's 25% chance, maybe it's still worth my while. But if it's less percent chance of that, or if worse comes to worse, I figure out that this cross has absolutely no snowball's chance in hell of getting me a platinum long-haired guinea pig, I'll be just wasting my time. That's the beauty of a cross. It allows me to predict an outcome, and I can decide to pursue the cross or not in real time if I think it's going to be worth my while. Let's take a look how a cross and a Punnett square actually work. How to do a monohybrid cross. Well, I've been talking about guinea pigs, so let's go ahead and use a guinea pig example. I'm not going to use anywhere near as exotic a color as platinum. I'm just going to use the standard colors for guinea pigs. What I remember when I was a kid, when my best friend Scott Gray headed off for Disneyland, which was in California. His family loaded up the car and they drove all the way from Pennsylvania to California to go to Disneyland. There was no Disney World in those days. And he let me take care of his lousy guinea pig. That was a traumatic experience. But the one thing I do remember, he's had one guinea pig that was black and one that was red. So I'm just going to appropriate the colors of those guinea pigs from when I was a 12-year-old kid taking care of them in his backyard. And I'm going to appropriate them for the purposes of demonstrating a monohybrid cross. We are examining only one trait in these guinea pigs: coat color. I'm not worried about coat length. I'm not worried about any other possible trait this lousy animal could have. I am going to go ahead and assume that maybe black coat color is dominant. It tends to be expressed. And red coat color is recessive. I will go ahead and use the letter A to designate the trait coat color. Uppercase A for black, the dominant. Lowercase a for red, the recessive. Now let's take a look at the two boxes I have down there. Each of those is a Punnett square. Let's take a look at the one on the left. Now the one on the left kind of shows me what would happen if I were to mate a homozygous dominant guinea pig with a homozygous recessive guinea pig. Take a look at the little designation below the Punnett square. You'll say big A, homozygous dominant. That is a black guinea pig. And a little X means I'm crossing that guinea pig with a homozygous recessive little a guinea pig, which would have red coat color. So what types of sperm am I going to make? What types of eggs am I going to make? Just for the sake of argument, let's suppose the big A is a male and the little a is a female. It doesn't have to be that way. It could be any way we want. But I need to figure out what types of sperm that male guinea pig's going to make. Well, I know through the process of meiosis, what Mendel called segregation, I can only put one of those A's into any one sperm in the process of meiosis. Well, since I've got two dominant alleles, I'm going to go ahead and put two dominant alleles on the left side of my Punnett square. My sperm will either contain the first capital A or contain the second capital A of the homologous pair. Same thing with the female. The female's going to make eggs, and she's got little a to contribute or the second little a to contribute. So I'm going to go ahead and list her egg types across the top. His sperm types on the left, her egg types across the top. Then I cross-index and I come up with the new combinations. A big A sperm fertilizes a little a egg, gives me a big A, little a genotype. That's heterozygous. As a matter of fact, every single combination results in that genotype. So I am looking at this Punnett square. I've got 4 boxes, 4 possible outcomes, but every single one of them is big A, little a, which means as far as genotype goes, you'll see the little question at the top of the two boxes, genotypic ratio. I've got a 100% chance of getting a heterozygous guinea pig. Phenotypic ratio, I've got a 100% chance of getting a black-haired guinea pig. Even if I mate a black-haired guinea pig with a red-haired guinea pig, I've got a 100% chance of getting a black-haired guinea pig in that next generation. So when they mate, there's absolutely no chance of getting a red-haired guinea pig at all. So let's take a look at a different variation. Now I'm now going to focus on the Punnett square on the right. And at the very bottom, you see my legend tells me I am mating two heterozygous guinea pigs. My male guinea pig might be big A, little a, and my female might also be big A, little a. Those are my genotypes. My big A, little a male is going to be a black guinea pig. My big A, little a female is going to be a black guinea pig. What sorts of sperm does he make? He can put the big A into a sperm cell, or he can put the little a into a sperm cell through an event of meiosis. What types of eggs is she going to make? Well, she can put the big A in an egg cell, or she can put the little a in an egg cell. So I will array his sperm types on the left. I will array her egg types across the top, and then I cross-index. What if I have a big A sperm fertilizes a big A egg? I get big A, big A. I got a homozygous dominant offspring. What happens if a big A sperm fertilizes a little a egg? I get a heterozygous offspring. So every single one of those squares contains a new combination. Now, every single one of those squares represents a 25% probability because this is one mating event. I'm mating two black-haired guinea pigs. And from that mating event, I've got four possible outcomes, 25% chance for each of those boxes. Now, work with me here. I've got a 25% chance of getting a homozygous dominant offspring. Big A, box in the upper left. I have a 25% chance of getting a homozygous recessive offspring, little a, lower right box. But then the other two boxes contain the heterozygous genotype. So I've got one box with big A, little a. I got another box with big A, little a. In my heart of hearts, at 25% each, add them together, that's a 50% probability I'm going to get a heterozygous guinea pig out of this mating. So, out of this mating, what's my genotypic ratio? My genotypic ratio at this point is the number of homozygous dominants compared to the number of heterozygous compared to the number of homozygous recessives. It would be 1 to 2 to 1. 1 big A to 2 big A, little a, to 1 little a. What about phenotypic ratio, Dr. Canasco? If I'm taking a look at this box here, this Punnett square, every time I see a uppercase A, that's telling me that's going to be a black guinea pig. So my upper left box contains a big A, my upper right box contains a big A, and my lower left box contains a big A. And then my lower right box contains the little a genotype. So at this point, I could probably say that my phenotypic ratio is 3 to 1. 3 black offspring to 1 red offspring as far as an outcome, or 75% chance of getting a black guinea pig versus a 25% chance of getting a red-haired guinea pig. Now, make sure that you understand that when we do a cross and we get a Punnett square, that Punnett square is showing us probability. These are not absolute numbers. And you realize, of course, that probability is just an indication of what your chances are. So, for example, on our last Punnett square, phenotypic ratio was 3-to-1. I could make these two guinea pigs over and over and over again, and I could always get a red-haired guinea pig. Now, that is in defiance of the probability, but you guys know that you can flip a coin 10 times. Theoretically, you ought to come up 5 heads, 5 tails, but that is very rarely the case. Generally, you wind up having a few more, maybe a few less, and it's entirely possible to flip a coin 10 times and get heads every single time. So our Punnett square shows us a probability. It shows us what our chances are in getting a particular phenotype or getting a particular genotype in an offspring from one mating event. Okay, it's time for us to look at a dihybrid cross. Now remember, this is a case where we examine the inheritance of 2 traits. In this case, we'll look at guinea pigs again, and we've got coat color symbolized by the letter A, uppercase A for the dominant, black coat color, lowercase a for the recessive, red coat color, and we'll couple that with another trait, hair length. I've got short-haired guinea pigs, which are dominant, symbolized by the uppercase B, and I've got long-haired guinea pigs, and that allele symbolized by lowercase b. What I'm going to do is I'm going to tell you this will probably not be something that you need to do on an exam, but I do need you to understand how a dihybrid cross works and see that the ratios come out just a little bit differently than they do in a monohybrid cross, but the theory is the same. So make sure that you understand how it works. I'm not going to make you do it, but make sure you understand how it works, because some of you are going to wind up in a genetics class at some point, and while you might not need it here, you definitely will need it then. So it might be good to get it under your hat at this point. Take a look at the left-hand side of our slide. I've got my parental types there. I've got one parent, which is heterozygous for both traits, big A, little a, big B, little b, and the other parent also heterozygous for both traits, big A, big B, little b. If I was to describe the genotype of those, they would be heterozygous. If I was to describe the phenotype for those, they would both be black-haired, short-haired guinea pigs, because the dominant allele is present in both of them. But let's take a look at the cross. Now remember, the first thing we've got to do is figure out what sorts of eggs or what sorts of sperm they're going to be making. Well, let's assume we start with a female this time. Doesn't matter, but the female is big A, big B, little b, and we'll put her egg types across the top this time. So what I got to do is, understanding that an egg contains only one allele of each gene, I need to go through her genotype, big A, big B, little b, and come up with all possible combinations of A and B, because her eggs is going to contain only one A and one B. So the combinations are, take the first A, which is a big A, and the first B, which is a big B, and I put that in my first column. I take my first A, which is big A, and the second B, which is a little b, and I put that in the second column. I take my second A and my first B, little a, big B, put it in the third column, and then wrap up with the last of the two, little a and little b, put it in the fourth column. So you see what I've done is taken all possible combinations of A and B from her genotype. And I'm going to do the same thing with him. Big A, big B, little b, and I see the sperm types are exactly the same. They're all possible paired combinations. Once I do that, what I do is I'm going to start filling in the new combinations. As I fill in new combinations, I always keep the letters grouped. So all the A's together, all the B's together. And when there's a dominant, that goes first. So if you just follow that idea, you can always keep things straight, because every time you glance at a box, it's always going to feature these letters in the same basic order, and you can very quickly determine what's going on. So I look at my cross-indexing here. If I have a big A, big B sperm, and I fertilize a big A, big B egg, I'm going to have a homozygous dominant zygote for both traits. If I've got a big A, little b sperm, find it over there on the left, and I'm going to fertilize a big A, big B egg, the second box down on my first column says I'm going to have a big A, big B, little b zygote. If I have a little b egg and I'm fertilize that with a little b sperm, I get a, little b homozygous recessive zygote. So then what I could do is I could go in and plug through exactly what the different phenotypes are. All right, well, let's see, what are the possible phenotypes? Well, I could have a black short-haired guinea pig, just like the parent. I could have a black long-haired guinea pig. I could have a red short-haired guinea pig, or I could have a red long-haired guinea pig. Those are my 4 possible phenotypes. Well, if I was to have a black short-haired guinea pig, I would want to look for every single box that had a capital A and a capital B. Now, work with me here, and you can go ahead and count them. There's 9 of them. There's 9 different boxes that contain both a uppercase A and an uppercase B, meaning that would be a black short-haired guinea pig. How many are going to be black with long hair? I'm looking for boxes that contain a dominant A allele, and I'm looking for boxes that contain a recessive B allele. And it's going to have to be little b. Can't be anything else. So if I'm looking for uppercase A and looking for little b, I find 3. So the other possibility is, what if I have a red-haired, short-haired offspring? Well, in this case, I'm looking for little a, and some big B combination. And there's 3 of those. And then finally, the last phenotype is, how about a red offspring that has long hair? That's going to be little a, little b, and there's only 1. So the phenotypic ratio here is going to be my probability out of 16, which means I'm going to have 9 out of 16 black short. I'm going to have 3 out of 16 black long. I'm going to have 3 out of 16 red short, and 1 out of 16 red long. If I do the math, 9 divided by 16 gives me a 56% chance of having a black short-haired guinea pig. I've got a 19% chance of having a black long-haired guinea pig. I have a 19% chance of having a red short-haired guinea pig, and then finally I have a 6% probability of having a red long-haired guinea pig. So what I've done is I've taken those fractions, 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16, and I've converted them to percentages. So once again, you're not going to be asked to specifically do a dihybrid cross, but once again, it's probably in your best interest to know how it works, and definitely know that the probabilities are essentially 1 out of 16 for every single one of those boxes, or 6%. Whereas, remember, for a monohybrid cross, it was 25% because there were 4 possible outcomes in a monohybrid cross, but there's 16 possible outcomes here. Now we'll turn our attention back to monohybrid crosses to take a look at something that Mendel actually developed. And this thing that he developed is called the test cross. Now, what we understand is that genotype codes for phenotype. And in Mendel's version of dominance, which is called simple dominance, you might remember there are 2 possible genotypes that code for a dominant phenotype. So taking a look at our slide here, I see a purple flower. And that purple flower is given a genotype P dash. Now that first P is an uppercase P, because we know that the purple flower color is dominant. The dash indicates the second allele. We don't know what that second allele is. We do know that a purple flowered plant could be big P, homozygous dominant. Or we know that a purple flowered plant could be big P, heterozygous. Mendel was confronted with this fact. If he had a pea plant that was demonstrating a dominant phenotype, but he didn't know what the parents were, and he had no idea what that genotype was. He knew in his heart of hearts he could have a dominant unit factor paired with a recessive unit factor, or he could have two dominant unit factors. He didn't know. So the question is how do you resolve an unknown genotype? Is that dominant phenotype going to be coded for by a homozygous dominant, or is it going to be coded for by a heterozygous plant? So he understood that if he took that unknown plant and he mated it with a homozygous recessive, in this case a white flowered pea plant that I know is p is homozygous recessive, then if the progeny or the offspring wind up being all purple, it's probably because the unknown parent plant was probably homozygous dominant. Take a look at the Punnett square on the left. If the genotype of the purple flowered plant is big P, and the genotype of the white flowered plant is p, we see that every single one of the boxes in that Punnett square is heterozygous, which means I have a 100% probability that the offspring will be purple. Put another way, if I breed a big P purple plant with a little p white flower plant, I will never, ever, ever, never will I never have a white flowered progeny. On the other hand, if that purple flowered plant is big P, and I put those alleles on the left, big P, and the white flowered plant contributes p at the top, I see at that point 2 of my squares in the Punnett square are purple and the others are white, which means if I breed this unknown purple flowered plant with a white flowered plant, if I come up with just one white offspring, that means that purple flowered plant is big P, So make sure that you know the test cross, which is an invention of Mendel, is a technique to resolve an unknown genotype. It means you mate the unknown plant or animal, which has a dominant phenotype, with some homozygous recessive, and that way you can determine whether that unknown plant is homozygous dominant or heterozygous. Now, I promised you, once we figured out how to do it Punnett square, we were going to return to the notion of blood type. Blood typing is a really good way of making a first determination of paternity. What do you mean paternity, Canals? I mean, if some woman accuses some fella of fathering her child, that's grounds for what we call a paternity suit. And one of the easiest ways of getting after a paternity suit is to do a blood type of the man, a blood type of the woman, and a blood type of the child. And you can very easily determine the possibility that he might be the father. Now, there's one very important point I'm going to make before we do this, and that is blood typing can disprove paternity, but blood typing cannot prove paternity. That's a very important idea. Blood typing can disprove paternity, but it cannot prove it. Let's take a look at an example here. I've got a male who has type AB blood. I've got a female who's got type O blood. And this female accuses the AB male of fathering her child. Let's take a look at 3 possibilities. You'll see 3 bullet points down here. First one, what if the female presented a type O child. Is this male the father of the child? Well, if you would be presented with this on an exam, what you would have to do is devise your own Punnett square, much like I see it here on this particular slide. You see the father is AB, and I see his alleles over there on the left-hand side of the Punnett square. I superscript A and I superscript B. And we know that the allele A and the allele B are codominant, and so therefore he's type AB. On the other hand, mom, the female, is little I, little I. So I've got that arrayed on the top of the Punnett square on the left-hand side of the Punnett square, and I fill in all of the spaces, and I see that I have at least two squares with I superscript A, little I, meaning that offspring is going to have type A blood, and I see two squares having I superscript B, lowercase I, meaning I've got a 50% chance I'm going to have a child with type B blood. What if the female presented a type O child? Is this male the father of the child? Well, I'm looking at the Punnett square. If that child is type O, it's going to be little I, and I do not see a little I on that Punnett square. In order for him to be the father of that child, he would have to have at least one lowercase I to contribute. He does not. I mean, I guess I could suggest that he would have to be type A, that is I superscript A, little a, I, or he could be type B, I superscript B, lowercase I. But in this case, he's AB, so he cannot possibly be the father of this child. So in this event, judge would throw the case out because he is not the father of this child. Second bullet, what if the female presented a type A child? Is this male the father of the child? Well, look at the Punnett square, and you see we have had at least a 50% probability that the child could be I superscript A, lowercase I, which means a child with type A blood. So the male could be the father of the child, but then again, any male who's AB could be the father of this child. Any male that's type A could be the father of this child. So we can disprove paternity, bullet point number 1, but we cannot prove it, bullet point number 2. It doesn't have to be this guy. It could be the boy next door. It could be the pastor of the local church. It could be your ex-boyfriend. Any of those people. Third bullet. What if the female presented a type AB child? Is this male the father of the child? Now I want you to watch this. She is showing up at court with a child as type AB blood. Now on first glance, you take a look at that Punnett square and you realize he cannot be the father of the child because none of those squares are AB. But now think about it. This mother is type O. This mother has only lowercase I alleles to contribute. This child doesn't have any lowercase i's. This child is type AB. This child is I superscript A, I superscript B. The judge would look at this woman and say, where did you get this child? This child is not yours. In order for that child to be hers, it would have to have at least one lowercase I in its genotype. So you need to be careful of this, because if you get presented with a blood type problem, I might do something evil and devious and ask you something like that. So make sure you know how to do a blood type Punnett square. Following these 3 examples here, be able to determine if the father is not the father of the child, be able to determine if the father could possibly be the father of the child, or is this child the woman's child to begin with? It's kind of fun. In a slide or two, we will be talking about X-linked or sex-linked traits. But before we get into that idea, we really need to get a handle on what types of chromosomes we have in the human genome. We've got autosomes, and autosomes are non-sex chromosomes. They are chromosomes number 1 through 22, and the traits found on our autosomes are called autosomal traits. And those traits fall in two different categories. There are autosomal dominant traits, and there are autosomal recessive traits. Hold that thought. We're going to spend a little bit more time, though, talking about sex chromosomes. The sex chromosomes are our 23rd pair, and they can be distinctly different. We've got something called the X chromosome, and we've got something called the Y chromosome. X chromosomes are indicative of females, Y chromosomes are indicative of males. But we're going to get a handle on exactly what that means. Let's take a look at the X chromosome first. The X chromosome is a genetically rich chromosome. In other words, it contains a lot of content. Any trait found on the X chromosome is called an X-linked trait. Any trait on the X chromosome is ultimately going to be connected to gender. On the X chromosome, we've got over 1,000 working genes. Some of those genes have nothing to do with reproductive development. And you could actually make the statement that the X chromosome's genes can be vital. In other words, the genes found on the X chromosome can be absolutely necessary to sustain life. Why, Canals? Well, because every single human being has at least one X chromosome. If you're female, you got two. And many times if you're female, one of those X's is inactivated. It becomes something called a bar body. And in males, you have at least one. Matter of fact, if you're a guy, your X is inherited from your mom. You inherit your Y from your dad. So every single human being has an X chromosome, which means that those chromosomes could have genes that are absolutely necessary for survival. Now, here's something interesting. The X chromosome contains the genes that essentially provide the blueprint for female reproductive development. Now, you might be thinking, why would the genes for female reproductive development need to be in all human beings? Because, Canals, you just told me that an X chromosome is found in males as well as females. Why would males need female reproductive development genes? Well, surprise, surprise, it turns out that the male reproductive development is based on female reproductive development. So guys are essentially female with a slight twist. Let's take a look at the Y chromosome. The Y chromosome is completely and totally unnecessary, at least for sustaining life. It's got about 86 working genes. None of them can be considered to be vital because essentially females don't have a Y chromosome and they seem to do just fine. Males on that Y chromosome do have a selection of genes that are collectively referred to as the SRY gene, which is really nothing more than a region, a set of genes on the Y chromosome that guide reproductive development towards being a male. So the SRY gene doesn't necessarily code for being a male, but what it does is it influences the genes for female gonad development and moves that development into a direction towards being a male. So one other bullet point here is that there is absolutely no crossing over between the X and Y chromosome during prophase 1 of meiosis. Every single other homologous pair demonstrates crossing over. You'll remember that's where the chromatids overlap. We got synapsis going on, we got breakage, and we got reattachment, and we got the shuffling of genes. That does not happen with the sex chromosomes X and Y because you couldn't possibly shuffle genes because you don't want necessary or vital genes showing up on the Y chromosome. It's not going to happen. So make sure you're aware of what the autosomes are. Make sure you know what the sex chromosomes are and the basic differences between the X chromosome and the Y chromosome. Not too long ago, in the context of talking about Mendel's laws, in particular his law of independent assortment, we mentioned about linkage. We said that if you have alleles found on the same chromosome, wherever that chromosome moves, those alleles tend to move together. They are said to be linked, or they are said to belong to a linkage group. Well, scientists have determined that if you have alleles located in certain locations on a chromosome, they might tend to stay together or they might tend to be split apart through the process of crossing over during prophase 1 of meiosis. See, if you've got breakage and reattachment going on, then you could have recombination going on with those genes on that particular chromosome. Now, let's take a look at our bullet points. Linkage. Alleles that are on the same chromosome tend to be inherited together. They are said to be linked, or they belong to a linkage group. Alleles that are closer together on a chromosome will tend to be inherited together, more so than alleles that are further apart. Increasing distance between alleles means an increased incidence of crossing over. There's an increased chance that they're going to be separated through the crossing over event. And those are described statistically as recombination frequencies. In other words, what is the probability, what is the likelihood that two alleles will be separated through an event of crossing over? We're going to see this on our next slide. This slide, shamelessly stolen from a website, basically reinforces what I was just saying. Recombination frequencies can be used to construct a linkage map of the chromosome. Remember, recombination frequencies are the probability that two alleles might be separated. Since the farther apart genes are, the more likely they are to be separated during crossing over, and the higher their frequency of recombination. Let's take a look at the illustration. I've got three alleles, B, CN, and VG, all located on the same chromosome. You'll see there is a 9% recombination frequency between B and CN and a 9.5 recombination frequency between CN and VG. In other words, there's a 9% to 9.5% probability that those genes will be separated from one another due to a crossing over event. But let's take a look at allele B and allele VG. They're further apart from each other. And you'll see all of a sudden their recombination frequency is 17%. There's an increased likelihood they're going to separate because they are further apart. So in the language of genetics, recombination frequencies are sometimes also referred to as map units, and they have been used as a way of locating the specific place of genes on a particular chromosome, what we call loci. Let's focus on X-linked traits. We know already that if you have a gene located on the X chromosome, it is described as an X-linked trait. Wherever the chromosome moves, that trait will move as well. And a lot of times, it's going to be associated with gender. So we're going to denote the presence of a gene on the X chromosome as X asterisk, as opposed to an X that doesn't have the gene. Now, if you have an X asterisk and you're a male, it will be expressed. Because the thing is there's no other X to mask over that allele. So if you see all males with the X and you see the genotype X asterisk Y, the X asterisk is the only X chromosome there. That asterisk allele will be expressed. On the other hand, if you're a female and you happen to inherit that asterisk allele on both of your X's, then you will express the gene as well. Bottom line, an X-linked trait will only be expressed in males if they inherit it on their X chromosome, and will only be inherited in homozygous females if they inherit on both of their X's. Now, on the other hand, there are females that are described as being carriers, and a carrier female is a heterozygous female. So I see she's X, X asterisk, or we could write her as X asterisk X. In any event, only one of her X chromosomes has that trait. Now, phenotypically, she's going to be normal. Phenotypically, she will not express that trait, but it's present on one of her X chromosomes, which means she can pass it on to future generations. Hence, she is a carrier. She's phenotypically normal, but she carries that gene on her X chromosome. So if we're taking a look at a cross-section of the human population, we've got normal females, that is, females who have the genotype XX, no sex-linked traits at all. We have a normal female carrier where she is a phenotypically normal female, but has the trait on one of her X chromosomes. She could be a female that is homozygous for the trait, found on both of her X's, in which case she is going to express the trait. We've got normal males who have an X without the trait and therefore are going to be phenotypically normal, or we can have males with the trait. They have an X asterisk, and we know that has been inherited from his mom. Now, can I give you an example of an X-linked trait that we could actually superimpose on this slide? So maybe I'd better understand it, Dr. Knaus. Yes, let's talk about colorblindness. Colorblindness is an X-linked trait. Colorblindness is the inability to distinguish certain colors, primarily red and green. And colorblindness is a lot more common in guys than it is in females. So let's take a look at this. The colorblindness allele would be found on the X chromosome. So we'll denote that as X asterisk. It is expressed in all males that happen to have that genotype X asterisk Y. In your heart of hearts, you got to know that that male has inherited that X chromosome from his mom. In other words, he got the colorblindness allele from his mother. Colorblindness is also going to be expressed in homozygous females. In order to be a female who's colorblind, you have to have the colorblindness allele on both of your X chromosomes. That's going to be pretty darn unlikely. So as a matter of fact, it's a very, very rare situation to have a female who is colorblind. A female can be a carrier, in which case she is a normal visioned female, but she's heterozygous. She's normal vision because she has a normal X, but she's a carrier because she's got the colorblindness allele on her second X. And we could denote that as X asterisk, or I could denote it as X asterisk X. Doesn't matter, as long as one of those X chromosomes has that trait. So if I take a step back and I look at all of the possible genotypes in the population, I got normal females, I got normal females who are carriers, I got females who demonstrate colorblindness, I've got normal males, and I've got males that demonstrate colorblindness. That is the trait on their X chromosome that is inherited from their mom. So what we're going to do on our next slide is going to take a look at some Punnett squares that examine how we might process the inheritance of a colorblindness gene. I've got two examples on this particular slide, and you might want to pause the video just to make sure you can jot some stuff down, because you take a look at the Punnett square on the left. I've got a female who's a carrier. She's X asterisk X. She is going to marry a guy who is normal visioned XY. We're going to see what the probability is arising in their offspring from a single mating of this colorblindness allele. If you take a look at her, she can have X asterisk eggs or normal X eggs. He can make X sperm or Y sperm. We do the cross index thing, and you'll see there's an X asterisk X in the top left. That's a 25% probability of having a carrier female as a daughter. I see XX on the upper right. That means there's a 25% probability of having a phenotypically normal female with no color blindness allele. I've got a 25% chance of having a son down the bottom left who is colorblind, and I've got a 25% chance of having a son who's normal visioned on the bottom right. Now, what happens if we mate a normal female with a colorblind male? You see the colorblind male has the X asterisk, and he can contribute that to a sperm. On the other hand, the woman has normal X. Cross-index the thing, and I see there appears to be 2 boxes with a carrier genotype in them, meaning there is a 50% probability we're going to have a carrier female as an offspring, and I see, likewise, a 50% probability of having a phenotypically normal son. On our next couple of slides, we'll be looking at alterations of chromosome number and alterations of chromosome structure. And we'll be talking a little bit about how we assess genetic conditions in families. We'll talk about tools used in genetic counseling and assessment. Let's start off with the notion of polyploidy. Polyploidy is a situation where an organism might have multiple sets of chromosomes. As a rule, some number more than 2. So an individual could conceivably be triploid or tetraploid or hexaploid or octoploid based on the number of chromosomes found in the individual. Now, this situation is more common in plants and is definitely rare in animals. See, for the most part, most sexually reproducing organisms are going to be diploid because diploid is the assumed result. Once you get a haploid sperm fusing with a haploid egg, you get a diploid individual. On the other hand, there are situations where you might have failures in meiosis, the failure of complete sets of chromosomes to separate, and that could set the stage for an individual that has multiple chromosomes. Now, the reason why it's more common in plants is plants seem to be a lot more tolerant of having multiple sets of chromosomes. On a practical level, having multiple sets of chromosomes means having multiple sets of genes, means producing larger amounts of protein. And in many cases, producing larger amounts of protein will have an improved productivity in the plant. It produces bigger fruits, it grows bigger. There are certain things that benefit the plant by having multiple sets of chromosomes. As a consequence, it sometimes has been selected for. And there are varieties and species of plants out there that do have multiple sets of chromosomes. In addition to being a natural phenomenon in plants, it actually has been bred into some crop plants. So, for example, bananas currently are primarily triploid. Wheat that is found in breads and in cereals a lot of times is hexaploid. And those big giant strawberries you buy at Walmart, the ones that are like the size of a baseball, those strawberries are octoploid. The normal size strawberry, that is a diploid wild type strawberry, might be about the size of a marble. But the big, huge, giant strawberries are a consequence of intentionally breeding polyploidy into that crop species. Now, I mentioned it's rare in animals. There are some animals that do demonstrate polyploidy, but by and large, not the case. Reason is because animals aren't anywhere near as tolerant of having multiple sets of chromosomes. Let's take a look at alterations of chromosome structure, a lot of times just occurring as happenstance results of DNA replication gone awry. There's deletions, duplications, inversions, and translocations. So got some illustrations here that's going to reinforce what we're about to say. A deletion is during an event of replication, you lose a section of DNA. So if you take a look at our chromosome on the left and it's been deleted on the right, you see a significant part of that chromosome is missing. And a deletion is going to have significant consequences because you're missing genes that could potentially make the difference between life and death of that cell. So deletion is a potentially lethal situation. Duplication is not quite so severe, perhaps. If you take a look at the duplication in this case, probably during an event of DNA replication during S phase of the cell cycle, instead of replicating a gene sequence once, we replicate it twice. And so I see two sequences of the same gene shown on the chromosome on the right. Now, if it's a problem making twice as much protein as before, then technically that could be a deleterious situation. On the other hand, if making twice as much protein is not, then it might be more benign than it is a negative situation. An inversion is when the order of the gene on the chromosome is reversed. So we took a look at that gene, and let's suppose on the left-hand side of number 3, the sequence would have read A, B, C, D, E. On the right-hand side, it would be E, D, C, B, A. Just switches around. And that might not be such a problem unless, of course, the reading frame has changed, which means you might be reading the gene backwards, so that's not going to make an appropriate protein. And then sometimes your controls might be changed. There might be an on-off switch next to that particular gene, and if the gene is switched, well, then the on-off switch might not be near where it needs to be. So it could potentially be a problematic situation, but much like number 2, could also be benign. Item number 4 is probably the most significant one. It's called a translocation. Now, it looks almost reminiscent of crossing over, but this is a case where we have breakage and reattachment occurring between non-homologous chromosomes. These are two completely different chromosomes. Let's suppose that the purple chromosome on the left is chromosome number 10, and the strawberry-colored chromosome on the right is chromosome number 15. And if we break off a piece of number 10 and exchange it with number 15, that could have significant problems as well, because you've got a whole set of genes on a completely different chromosome, and that could have disastrous consequences during a crossing over event, because conceivably you could have a chromosome arising in a gamete that might be missing genes or may have extra genes. Of course, that's going to have its implication after fertilization event, because you could then have an offspring that is missing genes or having extra genes. And once again, that could be a benign situation, but it could also spell disaster and be a lethal situation for that zygote. Now we're going to turn our attention to 3 basic types of human genetics disorders: recessive autosomal disorders, and those are disorders that are encoded in recessive alleles on chromosomes number 1 through 22, dominant disorders that are likewise encoded on alleles on chromosomes 1 through 22, but these are dominant alleles, and then finally X-linked disorders, which are encoded on the X chromosome. Now, each of these cases, what we are going to do is learn how we can diagnose these disorders by looking at a family history, what is commonly called a pedigree chart. Pedigree charts are fairly straightforward, and there's really only a few things you need to know about the diagramming process and the symbols involved. What you need to do is you need to be aware of the fact that males are always indicated with a square, females always indicated with a circle. If the individual is affected, that demonstrates the phenotype of that allele, whether it is autosomal recessive or autosomal dominant or an X-linked trait. That individual is going to be a shaded symbol, a solid square for a male, a solid circle for a female, and if the individual is otherwise unaffected, they will essentially be a empty square or an empty circle. That's all you really need to know. The rest of the details on this particular slide are not critical. The following 3 slides will document these different types of alleles in terms of how they appear in a pedigree chart. In each of these cases, I'm going to run through them fairly quickly, but you will see the pedigree chart illustrated showing you males and females and affected individuals and unaffected individuals. And on the right of each slide is going to be a list of kind of clues, distinguishing features, patterns, things you need to be aware of that you can look at. And if you look at those items on the right-hand side and compare them to what you see in the pedigree chart, you can pretty effectively determine what sort of disorder is making itself known in that family tree. So let's take this first one. The first one is an autosomal dominant disorder. So in this case, you've got an allele that expresses as a dominant. In simple dominance, you'll remember if the allele is present, it will be expressed. So in this case, every affected individual is going to have an affected parent, which means the affected individual has a dominant who's inherited that dominant from the parent. Generations are not skipped because, as a rule, dominants tend to be expressed, and when they do show up, they will probably show up in every single generation. And then finally, males and females are equally affected, which would automatically discount an X-linked disorder. If you take a look at a pedigree chart and it looks like males and females are equally affected, it's going to either be an autosomal dominant or an autosomal recessive. So on our next slide, we're going to take a look at an autosomal recessive and compare it to what we see here. Okay, well, let's take a look at this autosomal recessive. Affected individuals can have unaffected progeny because the fact of the matter is you could wind up having a progeny inheriting a recessive, but at the same time inheriting a dominant from the other parent, which means they would not necessarily express the autosomal recessive trait. But at that point, they would be a carrier. Generations can be skipped because you would have to have a homozygous recessive in order to essentially express the trait. Okay, and item number 3, if 2 mating individuals are affected, then all progeny will be affected because they have only homozygous recessive genotypes and there's only recessive alleles to be passed on to their next generation of offspring. Finally, you should probably add a number 4 here that probably states that males and females are equally affected. Once again, that discounts an X-linked disorder, but if I see these characteristics, this pedigree chart represents an autosomal recessive disorder. Alrighty, well, here's our third pedigree chart, and you'll notice the symbolism is slightly different. What the chart developer decided to do was to symbolize carriers. Now, you remember in a sex-linked trait, heterozygous females, that is, females who have both the normal X chromosome and an affected X chromosome, technically are carriers. They do not demonstrate the X-linked trait, like colorblindness, but they can pass it on. So in this case, a female who is represented by a half-shaded circle is a phenotypically normal female, but one who has the X-linked trait and can pass it on to future generations. So let's take a look at this. In an X-linked recessive trait, all X-linked traits are recessive. Affected individuals can have unaffected progeny. Generations can be skipped because essentially at that point the allele is going underground, at least in these phenotypically normal females. And males are going to be mostly affected because if you're a male and you inherit it on your only X chromosome, you will demonstrate that trait. Affected females are relatively rare because in order to be a female and express the trait, it has to be present on both X chromosomes. A pedigree chart is a relatively easy and definitely non-invasive way of assessing an individual's genetic predisposition to have a genetic disorder or to perhaps pass that allele on to an offspring. So, but what we're going to do is we're going to take a look at some of the actual tests that can be done to assess human genetic condition. A lot of times these tests are done on an unborn child in the uterus. As a consequence, you can actually determine if there is a genetic disorder before the child is actually born. So let's take a look at some of these tests. The first is the most basic. It's called a karyotype. Karyotyping is taking a sample of a cell, generally from the amniotic fluid or the placenta, and that cell is technically belonging to the baby. It's not mom's, it's the baby's. And it's an invasive test, but the fact of the matter is if you can obtain that cell and then encourage it to divide and then stop the division, you can actually take a photograph of the condensed chromosomes and arrange those chromosomes in order according to number, and you can actually determine if there are extra copies or missing copies, or if there's obvious damage to any one of those chromosomes. So to the right-hand side of our bullet points, you will see a photograph of a typical karyotype, which happens to be a karyotype for a male because you'll see one X chromosome and one Y chromosome, but all the other autosomes are paired. And I see that every single pair is complete, and there doesn't seem to be any problems with any of those. Now, if you wanted to, you could conceivably Google search for karyotypes and find some karyotypes, for example, of Down syndrome or Turner syndrome or Klinefelter syndrome or any other of these genetic disorders that you might be interested in. And you'll be able to visualize exactly what the problems are by looking at the karyotype. So karyotype is an important test in obtaining a cell, encouraging it to divide, taking photographs of the chromosomes, and arranging them. How do we obtain the cell? Well, there's two ways, and I mentioned they're invasive. There are two ways of doing this. In the late 1960s, something called amniocentesis was developed, and that is actually, in many ways, the preferred method. A bit later in pregnancy, when a woman's maybe about 15 weeks into her pregnancy, a sample of amniotic fluid is removed from the uterus. That amniotic fluid is the fluid that the baby is suspended in. That amniotic fluid will contain some of baby's cells. Through the use of an ultrasound, a needle is inserted into the woman's abdomen and a small sample of the amniotic fluid is removed. There is a very, very slight risk of miscarriage in doing it. But the fact of the matter is, if you can remove those cells, and at that point you can stimulate the cells to divide and do a karyotype. So amniocentesis, invasive, 15 weeks into the pregnancy. Early on in the history of amniocentesis, researchers were searching for some other way of maybe being able to assess genes earlier in the pregnancy, and they came up with something called the chorionic villus sampling method. In this case, what they're going to do is they're going to take a small bit of tissue sample from the chorion, which is part of the placenta. It's early enough in the pregnancy that there is not a significant accumulation of amniotic fluid, and that would be virtually impossible. So I say earlier in the pregnancy, meaning about 11 weeks. A lot of women don't know they're pregnant until they're about 4 or 5 weeks along. So technically, this is fairly early on to detect some kind of issue. A sample is taken from the chorion of the placenta. That will contain cells we can stimulate to divide and basically also obtain a karyotype. Now, I will tell you this. The chorionic villus sampling method has a slightly higher risk associated with it, and it has also been demonstrated to have some effect on limb deformities in children after they're born. So that any individual being encouraged to do an amniocentesis or a chorionic villus sample, and it is done, it is encouraged, needs to weigh the risks versus the benefits. Obviously, the benefits is you understand whether your child is going to have an issue or not, and that for many people means a decision to abort, or for others it's a way of being prepared. There's one other way of assessing for genetic disorders, and that is looking for the proteins that are generated from a gene that is considered to be a disordered gene. Genes code for proteins. So if you are harboring an allele that might code for some kind of genetic disorder, that gene is going to be responsible for making certain proteins, and those proteins are collectively referred to as genetic markers. So a blood test for the presence of certain proteins can determine, for example, if a man or a woman is harboring an allele that may conceivably pass on to a future offspring.